Method of identification of microorganisms, staining properties and biochemical test. Bacteria is divided into groups depending upon their structure of their cell walls, and it is reflected by their staining properties. There are three types of stainings mainly: simple stain, differential stain and special stain. In simple stain, which is used as a single basic dye such as crystal violet or methylene blue or safranin, etc. All bacteria take up the basic dye uniformly and appears in the same color. Only the morphology of the bacteria can be appreciated upon the staining. And the next one is differential staining. Here, gram stain is a method of staining classified gram-positive and gram-negative bacteria. The name comes from a Danish bacteriologist Hans Christian Gram, who developed this technique in 1884. If bacteria is treated with this solution, crystal violet and iodine, the cell stain purple. The stained cell is treated with a solvent such as an alcohol or an acetone or a mixture of these two. Some species like gram-positive, which is retain the stain, whereas the other, which is a gram-negative, which is loses the stain. The differential staining is attributable to the quite different molecular architecture of gram-positive and gram-negative cell walls. A gram stain can be easily and rapidly performed. It is important therapeutically because gram-positive and gram-negative bacteria have a differing susceptibility to a variety of antibiotics. Therefore, gram stain is used to guide therapy until definite identification of microorganisms can be obtained. Acid-fast stain. Most bacteria that have been stained with carbofugine can be decolorized by washing with an acetic alcohol. Certain bacteria which retain this carbofugine stain after washing with an acidic solution, such bacteria is called as acid-fast. Typically, they have a waxy material, mycolic acid, in their cell walls. Ciel-Nielsen staining is a type of acid-fast stain which is first introduced by Paul Ehrlich. Ciel-Nielsen staining is a bacteriologically stain which is used to identify acid-fast organisms, mainly mycobacteria. The most clinically important acid-fast bacteria are Mycobacterium tuberculosis and Mycobacterium leprae. Counter stain. In the gram stain and acid-fast staining procedures, the bacterial cell may be re-entered invisible by the decolorization step. Visibility can be restored by using a counter stain that has a color distinctly different from the primary stain. For example, in gram staining procedure, the pink dye safranin is used as a counter stain. Therefore, gram-positive cells are purple and gram-negative cells are pink. Special staining is a negative stain, which is a specimen or a part such as capsule resist takes up a stain. Here, background around the organism is filled with a stain such as Indian ink or an acidic dye such as nigrosin. This process leaves the organism cells as a clear, unstained object. Flagella stain is also a special stain. It is a painstakingly prepared to coat the surface of flagella with a dye or a metal such as silver. This technique is very difficult and time-consuming. Endospore stain. A few types of bacteria produce resistant cell called endospore. Their cell walls are resistant to penetration of ordinary stain. When simple stain used the spore seen as a clear and glassy. Identification of microorganisms by biochemical test. Voigt's Prosecute test or VP test. To differentiate the E. coli and E. aerogens by the production of 2,3-butadiene and acetoin via glucose fermentation. Development of crimson red color indicate positive test for E. aerogen and no color change indicate negative test. Pyrase test to differentiate Enterococcus species from non-Enterococcus species, useful for the presumptive identification of group A beta hemolytic streptococcus. Positive pink to cherry red color after addition of color developer. Negative, no color change in inoculated portion of the disc. Hippurate hydrolysis test to determine the ability of the organism to produce hippuricase, which hydrolyzes the substrate hippurate. Useful in the identification of Streptococcus agalacete, Campylobacter jejuni. Positive deep purple color, negative slightly yellow pink to colorless. Methyl red test. To carry out methyl red test to determine the fermentation capacity of a given bacterial culture. Methyl red is indicator that detects the pH after the Entity gram-negative rod has fermented glucose to completion. Positive, bright red color, and weakly positive is red-orange color and negative is a yellow color. Thank you.